is all yours. Thank you. Oh, wait, sorry, everyone. Those that have just joined us, please assess us with using the rubric to assess the student. As many of you as possible, please. Thank you. She's all yours. Thank you.
Thank you. Thank you, Adele. Good morning, everybody. My name is Marisha Chaulagain, and my supervisor is Chen Chen Zhao. And today I'm going to do a presentation on physiological and molecular response of barley to waterlogging. And it is funded, and the funding source is Grain Research and Development Corporation. So with global climate change, we can see that there's going to be increased temperature. As we can see in the globe here, the model, we can see that with increase in time, the world climatic temperature has been increased. And because of the increased climate temperature, waterlogging frequency has also been increased with increased time, as we can see from this graph here. So because of these global climate change, we are going to face extreme climatic events such as cyclone, high rainfall, which all leads to increased lateral groundwater flow, flooding, and increased groundwater table. All of this which leads to waterlogging stress for agriculture crops. And that brings in question the sustainability of agriculture products, so which we are going to address in this presentation. So why waterlogging? So in Russia, about 10% of the soil is prone to waterlogging. In USA, approximately 16% of the soils are in threat of waterlogging. And if waterlogging, or when waterlogging happens, as shown in the picture here, Australia faces a loss of 300 million Australian dollars. And to be more precise, cereal crops like grain crops has a loss of about $180 million. And in different states of Australia, such as in Western Australia, about 1.8 million hectares of land is under waterlogging threat. And in Victoria, about 4 million hectares of land is under waterlogging threat. So that's why we had the main issue as waterlogging for our topic. So the question is, what happens during waterlogging? So during waterlogging, when the soil is saturated in water and there's deficiency of oxygen for plants, so the plants are forced to shift their metabolism from aerobic respiration to anaerobic respiration. So that means the production of ATP is decreased from 36 to 2 ATP per glucose molecule. And this also restrains root development and reduce nutrient uptake in plants. This all leads to a weak root system for plants, and this weak root system is observed in the shoot as well, as stunted growth, reduced photosynthesis, and ultimately lifted, and finally causing a huge yield loss. So during evolution, plants, they have developed certain defense mechanism against waterlogging. So there are majorly 3 defense mechanisms, one of which is parenchyma formation. So parenchymas are small empty spaces inside the tissue that is formed under hypoxic conditions that allows the passage of oxygen from non-waterlogged areas to the root system. The second one is the formation of adventitious roots that is developed in the internodes, as we can see in the figure here, in the internodes on the hypocotyle or at the base of the stem. And these adventitious root has a number of parenchymas that facilitate respiration within the plants. And the third one is the radial oxygen loss barrier. So ROL is a phenomenon where oxygen is lost or escapes to the outside environment when being delivered to waterlogged areas. So ROL barrier prevents or stops the oxygen from escaping into the outside environment. So why did we choose barley? Because barley has been cultivated for a very long period of time in the world, with the production of 145.93 million metric tons last year. And also Australia, the barley is the second largest crop after wheat in Australia. And Australia contributes to about 12 million metric tons in this worldwide production. and half of which, more than half of the production is exported internationally for different purposes, and it contributes to about 30 to 40% for malting business and 20 to 30% as a feed. And because of, not because of waterlogging, but because of various reasons, and also one of which being waterlogging, barley production faced a loss of 1.7 billion last year. And more importantly, bad news for people who enjoys beer, it's gonna be an issue because beer prices are gonna go higher. So that's another issue. So the reasons for us doing this is because we do not know about the morphology and physiological adaptation in both shoot and roots. We want to understand the signaling networks between root and shoot, and we also want to have more of the genetic resources to produce good variety under waterlogging conditions. And also, we do not know the agronomic practices that alleviate waterlogging effects on barley. So coming to the research aims of my project, the research aim is to quantify the effects of waterlogging on crop growth reduction, to quantify the crop performance rescued by an parenchyma genetic loci under waterlogging, and also to have the information on waterlogging genes such as ERF and RBOH. So for the materials of my project, so we had two varieties: Planet, which is a commercial variety, good quality and good yield, but is waterlogging susceptible, and P52, which has waterlogging tolerant gene and is waterlogging tolerant. So we had two varieties replicated 4 times in 2 pots in the net house that is shown in the diagram, that is shown in the picture here. And we kept the seeds were sown at around late January and kept in the net house for about 3 months. And after 3 weeks of seed sowing, one of the pots was subjected to waterlogging condition for about a month. And then we collected data that included phenotypic investigations, and we also collected the data including stomatal physiology and morphology. We calculate the relative water loss rate and also the biomass of the plant, and we did some SPAD measurements, saw some stomatal physiological reaction, and also did the qPCR. And some of the equipments and the process that we did is shown in the picture in the slide. So coming to the results of the project, what we came to know is that waterlogging highly reduced plant height, leaf area, and root length. As we can see in the picture in the down in the figure in the down left, we can see that there is a significant difference in the treatment between P52 and Planet with plant height, but there is no significant difference between varieties. But in the root, as we can see in P52, we can see that P52 have performed much better than Planet in waterlogging condition because of the bulk that we can see in the root, and it explains why that is because of parenchyma genes that has helped the crop to survive under waterlogging condition. And also we can see that for the leaf area, the P52 maintained higher leaf area compared to that of the Planet. So we also came to know that waterlogging reduced photosynthesis capability through stomatal morphological traits, but not because of any chlorophyll content. As we can see, there is no significant difference in the top 2 figures on the top left, where there is no significant difference between the chlorophyll fluorescence and SPAD readings. And also there's no significant difference in the number of stomata between 2 treatments and varieties. But however, if we move to the bottom right, and near to the pictures of the stomata, we can see there the stomata area for P52 is higher than compared to the Planet, which explains that P52 maintaining higher photosynthesis capability. And also with the relative water content figure on the top right, we can see even though there is no significant difference at the very start, we can see at the end, the water loss in planet is higher compared to that of the P52. And these are also some of the picture at the bottom. I'm not quite sure if it's clear, but the stomata, the size of the stomata is comparatively higher in waterlogging P52 than that of the waterlogging planet conditions. So as we can see here, we also found that the waterlogged barley
So is it bothering you?
Yep.
Let me help. Let me help. Sorry, people, I just need to move this. What did I do? What did I do, Shane? I have caused, hey?
Yeah.
Oh, thank the Lord. Okay, all right. Is that good?
Yep. So waterlogging significantly reduced the photosynthesis, as we can see on the top left figure, that under low light, there was the photosynthesis rate of both planet and P52 under both treatments is very low. But under normal light, there is a significant difference in the photosynthesis rate between two treatments. But in stomatal conductance on the bottom right figure, we can see there's a significant difference in the treatment as well as between the variety at the bottom graph, where we can see that there's a difference in P52 and planet waterlogging conditions, which tells us that the decrease in photosynthesis rate is not because of the chloroplast or mesophyll content, but because of the stomatal morphological traits. I cannot click it.
Not working.
So next slide.
Oh. Hey? I'm clicking on, yep, there. Yep.
So for conclusion, I would like to tell that P52 indicated with all of the graphs and everything before, just trying to tell us that P52 indicated less reduction in root biomass and length. And waterlogging significantly reduced photosynthesis and biomass. And the decreased photosynthesis rate is more likely due to because of the stomatal conductance reduction. And for the future tasks, I still want to explore the content on the crosstalk between shoot and root to understand what happens under waterlogging if the roots are sending some signals for stomatal closures. It's still something that needs to be explored. And these are some of the reference. And at the very end, I would really like to acknowledge the help from my supervisor, Chen Chen, and also the help from PhD students, Abu Bakr and Anusha, who were very helpful and very kind to me during my stay at Launceston. And also, I would like to thank TIA for sponsoring the travel to Mount Pleasant. Thank you. Any questions?
Thank you, Marisha. Thank you so much. That's really appreciated. I must say, in the blow by blow, day in, day out work during the semester, I forget how interesting the work being done by our students, especially ATP, ADP energy, root energy. I know I should think about it, but I don't. So please, questions, any questions?
Yeah.
John, yes.
So the starting presentation that you had, you talked about three kind of hypotheses, how plants deal with waterlogging.
Yep.
So how do your results relate to that? So which of the three hypotheses or mechanisms are relevant to barley based on your results?
So all of the three are related to my thesis, because one is the formation of arenchymas. So arenchyma are basically formed, the formation of arenchymas are mainly governed by ERF genes. So that we are going to study in this research, and also the ROS. So ROS species, they are also governed by RBOH genes. So which also further we are looking into this thesis. However, I haven't done the analysis for the qPCR, which will tell us more about the ERF genes and RBOH genes, how they are working under that situation. So that is more yet to be done.
Thank you. Nathan?
Yeah, hi. Thanks for that talk. I just had a couple of questions about the whole concept of waterlogging. I thought your data was really interesting, but I don't quite understand how global warming leads to waterlogging. What's the connection there?
So I think I had said in the introduction as well. So global warming is gonna cause increased temperature and different other climatic events such as cyclone and rainfall. So heavy rainfall and like unclear weather patterns is gonna cause flooding, like increased groundwater table, everything. So all of this is gonna lead to waterlogging conditions for plants.
Beautiful. Thank you. Excellent answer. Any more questions, please? Thank you so much, Marisha. You've done, I was very impressed. Both of you, actually, both presentations. It'll be hard to take away marks from you, I'm afraid. Thank you. Thank you so much.
you.
Mr. Sadan Badu, please. I'll just, we'll close down Marisha's presentation and open up so we will share screen. Just getting it full screen. Yep. All right. So Sudan Padul, Supervisor Dr. Tori Clark, rapid assessment of nutrition value and performance of white clover in Tasmania. Please proceed.
Thank you. Thank you, Adam. Respected professors, researchers, friends. Good morning. Today, my project is rapid assessment of nutritional value and performance of white clover in Tasmania. This project supervisor, Dr. Tori Clark. So, begin with, I would like to proceed. What is pastures? Pastures is a forest that is grown as legume, mixed grasses, or hops. Pastures is given to the grazing animal, or also given by cutting them, or also saved for the futures as a hay. In Australia, we feed pastures for the beef productions, sheep, and lamb productions. So we need to have a pastures that have good quality for its productions, for meat productions or wool productions, to get healthy enough nutrition for the animal as well. If the animal get poor quality of pastures, then their meat production is not good. It will hamper their productions as well as the wool productions or lamb productions, having the less impact. There are a lot of pastures that are berseems, red clover, cowpea, but today I'm gonna focus about the white clover, that is Trifolium repens. It is a legume plant. It has high ability to fix the nitrogen. Also, it is high in minerals such as calcium, magnesium, phosphorus. Also, it has high protein, about 17 to 33%. Good quality of pastures is directly related to the good meat productions and also the milk productions. So there is a lot of things that changes these patterns or their value. There is a lot of abiotic and biotic factors that affect the low pastures productions, different stresses such as from biotic side, the insect disease, etc. And from abiotic, there is drought, waterlogging, rising temperature, etc. In futures, nowadays climate change is a very basic topic, and in futures there will be more intense. In the drought conditions, now from these stress, today I'm going to talk about the drought stress as well. So what is drought stress? Drought is the insufficient water level in the soil. So in drought conditions, what do plant do is they shut down their stomata, and all the primary functions such as photosynthesis and all, it will slow down, and eventually it will wilt, and then plant get died. When there is insufficient water, the whole photosynthesis parameters slow down, and plant does not respond well. Ultimately, they die, and also by eating or not getting enough food for the animals, they not gonna be perform well. So in order to manage and in order to management or the producing business breeding tool, my research is going to look, can we actually detect the early signs symptoms before visual indications? That is my main questions. Can we actually detect the early sign and symptoms before it's showing any visual signs? There is a lack of rapid assessment tool. If we took any measurements in the photosynthesis parameters, we gonna take only like 8 to 10 sample per day from gas analyzer. So from this, we also gonna develop some rapid assessments tools. Is that really work in a white clover or not? After knowing the early sign and symptoms, we're able to detect whether the plant need water or not. If they need water, we can immediately irrigate that so that they can regrow again. What we see now is like, after showing the symptoms, after they get wilt, if we water also, they will eventually die because they cannot recover it. So there is a traditional way for this measurement, but like it is not rapid. Also, it requires the dark conditions. First, we have to close the we have to take to the dark or the rapid and then take the measurements. But the tool which I'm talking about, that does not require any dark adaptations. And materials and methods. The experiment was conducted on the glasshouse of University of Tasmania, Burnie. I went Burnie there and stay about 12 to 13 days. And so seed was first grown on the filter paper and in the dark, and then transplanted to portraits. And after that, it will take to the glass house in controlled environment. After three weeks, the white clover seed set into the experimental plot. There were flow treatments, that is 100%, 80%, 65%, and free drain one. There is a series of experiments conducted to check whether it is working or not. But today I'm going to talk about the only two project. What is the field capacity? Field capacity is total water holding capacity of the soil. So how we determine that is we first wet the spot with the soil and then add free drain water, and then keeping the water free drain, we constantly wet that until it goes the constant wet. So that's how we find the field capacity of that particular soil. So why we choose these 100%, 80%, 65% to represent the moderate and severe drought conditions or controlled drought conditions as well. I used the tool called multi-spec for this measurement. The measurement was mainly taken in the morning, and by clipping the leaves of the clover, as a clover is trifoliate. So I always try to clip on the same leaf by marking the two sides, so that I always go and take on the one that particular leaves always. And photosynthesis writes 2.1, that is the protocol of the multi-spec, and leaves well measures every time by taking marking the two slides. And for the analysis, RStudio and Excel was used. And this is the tool called a multi-spec. It is a low cost developed by the Plantex, and then it is a rapid. It only takes like 15 seconds. If you click that, and it will flash the light, and it will detect all the parameters of photosynthesis parameters, such as Phi2, NPQT, that is non-photoquenching light, which I'm gonna talking about. And okay, before going to results, first I would like to tell what is photosynthesis and what happened in the photosynthesis. Photosynthesis is a very complex process. It is a transferring light energy into chemical energy. When the light energy strikes on the leaf, it directly it goes to three ways. First, it goes to photosystem 2, and then secondly, it re-emitted back, and thirdly, it dissipated as a heat. And after the heating in the leaves of leaves, then it will go to photosynthesis 2, having their photosynthesis, and it will still the proton have the maximum energy, so it have to re-emit it back as a chlorophyll fluorescence with a certain wavelengths that we can measure. The chlorophyll fluorescence, the principle of chlorophyll fluorescence is opposite of Phi2, that is photosynthesis 2. If inside the plant there is a maximum photosynthesis, then the chlorophyll fluorescence will be low. And if chlorophyll fluorescence is a high, then we detect that there is some kinds of things happening inside the photosynthesis. So my results, I try to maintain the 100% field capacity throughout the periods, and 80%, 65% throughout the period. But like in the day first, there is a significant difference between all the treatments. So I could not maintain the 100% fill capacity, because like there is a lot of things happening inside the glass house, like maximum there is a fluctuations in daylight temperatures, and also some days is humid, and like it's reflected directly to my results. So there is already significant difference between the field capacity in day 1, and in day 5 there is no significance difference. So result is not came as per expected. So we conducted series of experiment, not only that one. So also in the for Phi2, it's the efficiency of a photosynthesis 2. And there is an NPQT, is a non-photoquenching light. After having the photosynthesis, it will some of the light reflected back. That is called the NPQT. And in drought stress, it's gonna be more high, but the result we got is like there is no any significance difference between them. Then we again analyze another parameters, what is that affecting and how that affects. So we found that I try to maintain the 100% fill capacity control, and the 80% and the 65% by weighing and watering every day. But like daylight temperatures is too high, as for ambient temperatures 25-30 degrees, sometimes 26, and there's a humidity as well, sometimes rains, that gonna fluctuates my results. So even I've tried to put 100%, then it goes sometimes 40%, 60%. So there is not good results. And another experiment, that is water and non-water. I fully just like taking a 2 sample, water and non-waters, having a replications of 8 plant, and the result I get is like there is a significant difference between the plant, but I'm not sure it is due to the stress or not, because already when I put the experiment on the side, it shows a significant difference between. So learning from this is, it is very it is very hard to maintain my control stress. Maintaining control is too difficult, and also maybe this is due to this is not a proper way to do the drought stress. I can't get steady control some of stress, some of other stress such as heat can be done through this device. And also I would recommend try different methods for the further because the method I use is too variable in environmental conditions, and the result I get is too varies and problematic as well. And this is my reference. Lastly, I'll acknowledge the help of Dr. Corey and the PhD student Akim for the help, also TIA for the sponsoring to go Bernie and stay there. Thank you. Any question?
Thank you, Perul. Sudan, sorry. Sudan, thank you so much. Excellent. I'll just stop sharing. Yeah, so please stand here so that you're visible. Yeah, any questions, please? Yeah, John.
My question is, if we can detect drought stress early, what's the benefit of that? So what can you do if you detect drought stress early?
Yeah, if we detect the draft, thank you for the questions first. If we detect the draft stress, if the plant is having a drought stress, they're showing that their photosynthesis is low, then we can put early water so that the plant can regrow again. Yeah.
So it's useful in situations where you can provide water.
Yeah.
But when you can't, then it's not going to help you very much. But,
If we can't.
yeah. So it's useful when you have the option to irrigate.
Yeah.
Yeah.
Yeah, and like if we saw the sign and symptoms already, and the plant can't revive without even putting the water, and there is no use for that. Yeah, that's why we try to get an early sign and symptoms, warning signs, and like that's why we can do that.
Thank you.
Yeah.
So drought stress, this is clover, but if we're talking wheat and protein, it does actually affect the level of protein in the grain and the quality of the protein, and that has downstream effects on processing, bread making, pasta making, etc. So drought, and of course, the reverse waterlogging has a large effect. Any more questions, please? Questions? We can't let Sudan off too easily. I guess you will be let off easily. Thank you so much, Sudan, for your time. Appreciate it. All right.
Thank you.
Thank you. Madhav Sunar, please. Madhav, have you got a USB in your hand? Oh my Lord. Oh my Lord. We should have done this earlier. Yes. All right, excuse me, people. I apologize. That's Madhav. We'll have to close yours. Okay. Yeah, just excuse me, everyone. Just loading up Madhav's talk. I did have an earlier version, but he seems to have made some changes. So this, which one is it? Thank you. Yep, and we will go back and we will share screen. And yep, go here, share, and then go full screen. Have we gone full screen? Yep. Thank you so much. All yours.
Yeah. Thank you, Adele, for this stage, and thank you for fixing the technical issue. So hi, everyone. This is Madhav, and my supervisor is Saint Powell. And I'm here to present my exciting topic, which is optimizing the production of plant-based kefir in the lab. And I'm also grateful to the university itself and Unigrain and ARC because they approach for the research. So let's talk about the kefir. So with the increasing need of the functional food among the consumers, kefir is being popular because it is rich in probiotics. And we can see that the global kefir market value is almost around 3 billion in 2023. And with the increasing compound annual growth rate of around 6% between the year 2023 and 24. And in Australia, the popularity of kefir is increasing because it is acted as a fermented food. And my project will aim to address the following research questions. Can we replicate the dairy kefir with the help of plant-based milk? And can we optimize the production of plant-based milk or not? So let's dive into kefir itself. So what is kefir? So kefir is basically a fermented food, which is made from the fermentation of the milk with the help of either kefir grains or starter culture. And kefir grain itself is a powerhouse. So
Yeah, keep going, keep going.
So can I remove this?
Where's my, why isn't it charging, Shane?
Well, charger, isn't it?
No, please, yes.
You got a Dell.
Keep going, keep going.
Yep. And kefir grain itself is a powerhouse of different strain of bacteria and yeast, almost around 50 strain, which convert the milk sugar into lactic acid. And the major strain
Uh-oh, we lost them.
Yep.
Whoops.
So we just wait.
I think the computer battery might have gone flat.
I've just sent a text to my student who's there. So she's going to hopefully tell Adele if she hasn't realised.
Tori, while you're there.
Oh yeah, I was just writing back to you. Yeah, so
Okay. Yeah. All right.
hand watering.
There was hand watering.
So he was weighing the pot
Wow.
and then adding the water back by weight and doing that 2 or 3 times a day. But it was really variable with like evaporation. It didn't work.
Why didn't he just use a drip system?
So we don't have that set up in the glass house. Initially, we just did it where he just stopped watering one set of plants as a treatment, which actually worked much better.
Yeah.
Yeah.
Or so drip systems are super easy to set up. You can set them up for like $20. But
yeah, he was only here for like less than 2 weeks.
Oh, okay. And what about like a watering mat? A capillary mat?
Oh, like as in bottom watering and then going.
Yeah.
Yeah, we didn't try that, but that
Okay.
would be an option. Yeah, I don't really like drought stuff, so I don't want to do any more.
Okay, no worries.
Yeah, no, it
Could
was
you
very
please
rough.
turn off your speakers? Sorry, please, thank you. Share screen. And full screen there. All right, yep. She's all yours. Thank you. Sorry, apologies, everyone.
Thank you again.
Wait, well, are we recording? We are? Good. Okay, good.
Thank you again. So there are numerous health benefits associated with the kefir consumption. The first being it helps to improve the body fitness and homeostasis because it is rich in different vitamins and minerals. And second being it supports the healthy gut because it is a diverse form of microbiome. And along with that, it also helps in the bone density and the formation of new bone tissue. So, however, traditional dairy kefir faces multiple challenges. For instance, there are people who are lactose intolerant, vegan, and even dairy allergic. Not only that, it also contributes to carbon footprint. For instance, if we see in Australia, almost 1 in 50 infants are dairy allergic, while 1 in 25 are lactose intolerant. And also like in Australia, around 2.5 million people are vegans. And the dairy milk, it contributes around almost 3 times of greenhouse gas emission and 9 times of land use as compared to plant milk. That's why plant-based milk can offer that promising solution to this issue. So talking about the non-dairy kefir, we can't ignore the water-based, like water kefir, which is also known as TVCOS. And it is fermented with the, it is a sugary water which is fermented with the help of grains. But it is low in calorie intake and fat. So therefore, plant-based milk, which can offer similar taste with added nutrition, can be an alternative to that because it is providing additional nutrition. And not only that, the plant-based milk has similar health benefits. Along with, we can serve to the lactose intolerant and vegan population. So the growing prospect of the plant-based milk can be observed in here, like soy, almond, and coconut milk has been fortified with different strain of blue-green algae, and because of that, we can produce different vegan kefir. And there are limited resources which has been carried out in the field of optimization of plant-based milk into kefir. That's why this research is quite important and interesting. So to address those research questions, like we can replicate or not the dairy kefir by the help of plant-based milk, and whether it is possible to optimize the different plant-based milk, we come up with these objectives. And there are multiple objectives, but because of the time consideration, like I will only talk about the 2 major objectives in this presentation. They are like to investigate the feasibility of plant-based milk with the help of starter culture, and to understand the different effect of concentration of starter culture into the exopolysaccharide secretion or production and lactic acid production of the plant-based milk. So to meet these objectives, we conducted this experiment in the lab in completely randomized design with 4 replicates and with 3 different concentration. And it was used to measure the lactic acid and EPS, that is exopolysaccharide production, with the help of the software called RStudio. So for this research, we consider 4 different plants. They were almond, coconut, oat, and soya, and dairy as control. And the source of inoculum was starter culture, which was obtained from Green Living Australia. Thank you for them for providing that starter culture as a inoculum. And then the research was set up in a simple way, but with sophisticated analysis, which can be observed in the slide. That is like we first fermented the milk with the starter culture, and then it was fermented for around 48 hours at 19 degrees Celsius, which was room temperature. And we analyzed the different quality assessing parameters of the kefir, such as pH, lactic acid, exopolysaccharides, and microbial count. But in this presentation, I'll be only presenting my lactic acid and exopolysaccharide production, because these are the quality parameters that will help help us to understand what is the mouthfeel, viscosity, and flavor of the kefir we produce. So I'm going to consider these 2 major quality assessing parameters in this project for now. So for the pH measurement, I use accumate pH meter, and for the lactic acid we use G20 compact titrator, and we also did colony count and EPS measurement. So so the whole aim for this in this presentation will be talking about the two major things: whether we can ferment the plant-based milk or not with the help of starter culture, and how the different concentration will affect on the lactic acid production and EPS production, exopolysaccharide production of the kefir produced. So moving on to the first section, which is like to see the effect of whether the milk will be fermented with the help of starter culture. So we got these results, like in terms of the exopolysaccharide secretion or exopolysaccharide production. What we got was soy milk and the oat milk were quite significantly different than the other plant-based milk, which was almond and coconut. And then in case of lactic acid production, only soy milk showed like positive results, because it might be because, and we came into the conclusion that it might be because of the protein content in the soy milk, and the lower amount of the EPS and lactic acid production in terms of coconut was because of the higher fat and specific gravity. And while oat can still be one of the milk which we can use because it contains beta glucan, which helps in the enhancement of the fermentation. Moving on to the next section, which is like to see the effect of different concentration on different milk. So what we got was, within the treatment, there was no significant differences except one, that is high concentration of dairy and the low concentration of dairy. While between the treatments, obviously dairy will perform better because of the high nutritious content it has. But regarding the other plant-based milk, there was no significant difference. But the results which we got in here was like contradictory, because one of the studies shown that EPS production is quite high in soy milk as compared to dairy, which was inoculated by Streptococcus thermophilus. And regarding the lactic acid production, so as similar to the EPS production, there was no significant difference within the treatment, but between the treatment, only oat milk and dairy milk was like quite significant. And it was concluded that it might be because of the higher concentration, there will be higher number of microorganisms, resulting into the faster fermentation, and that produced the more lactic acid in dairy and oat. But in terms of oat milk, which we use in the research, it had like higher carbohydrate, but there was low protein content, which was around 3.8 gram. So which might restrict the production of lactic acid. We concluded that. So in conclusion, we can say that all plant-based milk can be fermented, but soy and oat are the viable alternatives because of the production of lactic acid and EPS. And regarding the soy milk, because it produces higher EPS and lactic acid, which can even be observed in here in the second picture, like oat milk is producing more lactic acid as compared to dairy milk, even though they are statistically not significant. So it can be like we can optimize the production of soy milk. And regarding the oat milk, although it showed like lower lactic acid and EPS production, like adding protein can be a like good addition to that for the fermentation, because it has like high amount of sugar but low amount of the protein as compared to soy and dairy milk. But there were a few limitations regarding the research which we conducted. The sample size, the replication number was quite low, which was 4. And then after the processing technique of different milk, which we use, the plant-based milk, might be different, which might create variation in the fermentation process and the production. And not only that, the starter culture which we use in here, provided from Green Living Australia, the microbial constituent was quite limited. So that might be another case which might change the production of lactic acid and EPS. So we recommend that we can use plant-based culture for fermenting the plant-based milk to get good results, because which we were using in this project was the animal one, the dairy one. And then after, we have to investigate more on the sensory properties of the plant-based milk, which we obtain here through the sensory panels. And further research will be carried out for the addition of more protein into the oat milk and addition of sugar and protein into the almond and coconut, because we have already got good results regarding the sorry for that. So regarding the almond and the coconut, we already got good results because of the fortification of blue-green algae. So if we can add more sugar and protein, that might give better results. So that is our future directives and recommendation. And finally, I would like to acknowledge my supervisor, Saint Powell, and Adol Yusuf and Caroline Clay for supporting me continuously. And I would also like to, I'm also grateful to Nadine, who provided us concentrate, who is working in Unigrain, and Ramandeep for giving me the research project work. And I would like to thank to the micro lab staff as well. Thank you all.
Thank you so much, Madhav. And thank you for acknowledging me. I didn't do much for you. It's the other staff that did all the work. So thank you very much. We'll stop sharing, and we'll call for questions. Please stand here. So thank you. Who's got his hand up? Nathan, please.
Yes. Hi. Well done, Madhav. I think I misunderstood your results maybe. So it seemed like none of the plant-based milks really they didn't produce the same amount of lactic acid as the dairy milk. But you were saying at the end that it's probably a viable alternative. Isn't the lactic acid important, like, parameter, kefir that?
Yeah, obviously lactic acid is the most important parameter, because it helps to inhibit the different different microbes which are inside the gut. And so that is the reason, yeah, we took this parameter into consideration. And regarding the plant-based milk, whether it produces more or not, in the first experiment, while we were comparing different concentration, like different plant-based milk, soya milk produce more lactic acid, obviously. And then in the second, yeah, it also produces more. That's the reason, like, I think, yeah, we can consider plant-based milk. Yeah.
Right. I must have just misread the slide. Sorry, my mistake.
Yeah, thank you. We've got a question from John here.
Got two questions.
Oh, lovely. Two questions.
What is backsloping? You said you used, what is that?
Yeah, backsloping, I use this method in my research, but I haven't presented here. Backsloping is basically what we do is, first of all, we ferment the milk with the starter culture. Then after, we use the fermented into the other fresh milk. So that is basically backsloping.
Okay, so it's like poultry.
Yeah, from the previous cultured.
Okay, and my second question is, did you taste any of them?
No, because we are quite concerned about the health. That's why I think we need sensory panels to know the sensory properties.
It's all about health, this product. So next time, maybe taste it.
Yeah, for sure.
Taste it first before you ask other people to taste it. Oh, yeah. Please.
Great job there, Madhav. That was a really good presentation. A lot of thought and about the importance of the project. So well done. Now, Madhav, my question, I think you sort of alluded to it, but you were trying to correlate why the plant-based kefirs could be better or not than others. And you suggested protein, and you suggested the high protein content of soya milk was the reason why it probably has the potential to be a kefir milk. But you didn't actually provide more information about what the protein levels were for the other plant-based milks. Can you just remember or give us an idea about roughly where that protein content is?
Yeah. So the milk which we use were brought from, purchased from Woolworths. And what I observed was in the ingredient, like in terms of, I can't recall the exact number, but in terms of the protein content, the oat milk had like 3.8 gram of protein, but it had like high carbohydrate concentration, which was 20 gram. 20 gram. But in terms of the dairy and the soya milk, it was around 8 gram. Dairy milk had 8.5 gram of the protein per serving, while soya milk had 8 gram of the protein.
So
I think-
almond and what was the other one? Coconut were really low protein.
Yeah, 0.3 gram. Yeah.
Okay, so you're not getting really
1.3
much
or
for
0.3.
those plant-based milks. So that's really interesting. Maybe you're drinking just water. All right, excellent. Thanks very much for answering that question. Well done.
Thank you. All right, round of applause for Madhav. Thank you so much for your time.
Thank you. Thanks.
All right. So our next speaker is Manisha Giri. Manisha, would you like to come up here? Well, Manisha's coming up here. Her supervisors are Dr. Sally Bound and Dr. Bianca Das. Manisha is going to talk to us about evaluation of oat hull ash on plant growth and soil properties. We'll get Manisha's presentation up. Yeah. You as well. Oh, okay.
I've sent you the revised version
Yeah.
early morning.
Yeah. So I've got the revised version. Yes, I've uploaded it.
Okay, then.
You happy?
Yeah.
I got it this morning. I saw it this morning. So, what number are you? Number 5, Manisha. So, yeah. So let's share screen and share, and then let's get rid of that and full screen. Yep, go ahead. Thank you, Manisha. Away you go.
Hello, everyone. Myself, Manisha Giri, and my supervisor as a Dr. Sally Bound and Dr. Bianca Das and Nadine as an industry partner. The topic of my presentation is evaluation of oat hull ash on plant growth and soil properties. Firstly, I would like to talk about what is oat hull ash and what are the problems associated with it. So, oats is really important for an Australian industry because Australia is the largest producer and exporter of both the raw oats and the processed oats. About 1.5 million metric tons of raw oats are produced each year. And during the commercial processing of oats, the raw oats, they are dehulled. And during the dehulling process, as a residue, oat hulls are obtained, and it consists 20% of total productions of the oats. And when they are used as in the biofuel, then the oat hull ash are generated, which is 75,000 tons per year. But till now, there is no any use of this oat hull ash, and they are going to the landfill. So I just want to know whether this can be used as a soil amendment or not. So now, I have, in many literature review, I found they are mainly focused on the rice husk ash, where they have demonstrated the use of different rates of OHA to improve the plant growth, but they have been only used in the limited crops like the cotton and maize, and the research they have been only taken in the tropical countries. So there is no any data for the subtropical or the temperate regions. Also, they have investigated the combined effect of rice husk as both the fertilizer and the soil ameliorant. So it is unclear whether the ash did provide the fertilizer effect or they help to ameliorate the soils. And talking about the oat hull ash, there's no any available informations. And also, we don't have idea whether it behaves similarly to rice husk ash or not. So my project aim is to identify the effect of oat hull ash on plant growth and development, soil chemical properties, And next is to determine whether the additional nitrogen is required or not. So the methods I follow during my experiment is I use the source water as from the unigrain mill, and I collected the acidic sandy soil which was under the seed pasture from the uniform, and I grew Dwarf sunflower as a model crop. And for treatment design, I use two-way factorial. First one is the OH rates by volume. I use the 4 rates. And second factorial is the nitrogen, which is plus in the minus. And for trial design, I have used the RCVD with the 5 replication in each treatment. And talking about the statistical analysis, I've used the t-test, one-way ANOVA, and the two-way ANOVA. I took the pre-harvest plant and the soil measurements, which includes plant height, node number, spot reading, and the flowering development. And I have done pH and EC by myself, but mineral analysis they have been done in the commercial lab. And now I will do the post-harvest measurements like leaf area and the root development, and again for the soil like pH, EC, mineral analysis, and the water infiltration rate. So this is my initial soils result. With the pH, I have done pH using the 2 methods. One is with the water, and next one is with the calcium chloride. So here, the soil, which is also considered as a control, the pH for that soil was 5.5, and which was acidic. And when you add the OHA, the pH has been increased, and the highest treatment, it's showing the highest pH, which is about the 8.5, and I think this is too high for the plant growth and development. And talking about the EC, the control, which is the soil, it was a sandy soil, so there were no lots of salts in it. So when you add the OHA in it, the EC has been increased. It may be due to the potassium content. I'll show that within my next slide. And so I think the treatment with this, all the treatment with this EC will not affect the plant growth. And this is my soil initial chemistry data. And on this data, on adding the OHA, we saw that decrease in organic carbon, increase in potassium, phosphorus, micronutrients, boron, and we don't see any major changes in nitrogen. Now next is the plant height. I took the measurements of plant height from the 14 days to 70 days. And when I looked at the effect of different rates of OHA, then I saw the significant difference from the 35 to 56 days, with 10% OHA showing the highest plant height compared to the other treatments. Likewise, the interactions, we can see the negative interactions of 50% OHA treatment with the nitrogen, because I have applied the nitrogen at the 21 days, and from the 35 days, the treatment 50% OHA with the nitrogen, they have declined their growth, and from 42 days they have died. They are almost dead. So talking about the interactions, we see only the interactions at the 35 days for all the treatments. Now, next is the number of nodes. So counting numbers of nodes is important because nodes may impact the flowering. So from the day 21, there is a steady increase in the number of nodes, that is up to the 9 nodes per plant in every treatment except the 50%. And we can see there are significant differences at the 28 days, 35 days, and the 42 days. And the difference at the 28 and 35 days is due to the slower growth at the higher rate. And at the 42 days, there is a slow growth in 25% of treatment. Now next is the flowering. So the graphs, it's showing the time take on the days to flower. When you talk about the sunflower plant, then we mostly consider, or the most people consider about the sunflower plant to open as soon as possible. So here we can see control is taking 63 days to flower, but we can't see any effect in 10% OHA. And in 25% OHA, we can see there is delay in flowering by 7 days, and in 50%, there is delay in flower nearly by 25 days. And the figure or the image on the right side is just the initial data where I'm showing the number of axillary flowers, and the treatment, 10% treatment with plus nitrogen is showing the highest number of axillary flower compared to the other one. So lastly, my conclusion on my first objective is the 10% OHA has helped to increase the plant height and the axillary flowers, where 25% and the 50% OHA, they have shown the negative effect on height, and they have delayed the flowering. And talking about the soil properties, we can see the strong alkalizing effect and also the slightly salinity effect. And they have also increased the available available potassium, phosphorus, and the micronutrients like the boron. So my recommendations for farmers is to suspect the stronger alkalizing effect than the rice husk ash, and only to use on the acidic soils. It is not recommended for the neutral soils, but you can use AGI to remediate the strongly acidic soils. And I recommend the 10% OHA, but for that, the plants should be alkaline tolerance or the preference. And lastly, there should be there should be a cautions against the nitrogen applications with the higher rate of OHA, because I have observed the negative effect when I apply the nitrogen with the 50% of OHA. It may be because my soil, it already has the high nitrogen, and on adding the nitrogen to it, it has screwed the plant in the alkaline environment. And lastly, I would like to acknowledge all the people who helped me along this way, because this study was really challenging than I initially expected. So there was one moment when I applied nitrogen and I was worried that my all plants is about to dead, but I'm excited to report that they have provided the valuable results today. So thank you, and thank you everyone for your attentions. And any feedback and suggestion so that I can improve something in my upcoming thesis writing. Thank you all.
So thank you so much. That was very interesting. I certainly, my soil chemistry is very bad, but I certainly have some questions. Alida, oh, did you have your hand up, Alida, or maybe not?
No, I can ask a question. That's fine. But I was just a clap. But thank you, Manisha. That was a really good effort. Some interesting results there. Now, Manisha, I just wanted to clarify. I may have missed it, but when you applied the oats to the soil, how long did you leave the oats in the soil before you put the seed in? I just want to know, so the incubation period, was there time for the oats to actually break down into the soil, or that's my question, sorry.
Firstly, I germinated the sunflower seeds in the peat pellets. Then only after I transplant into the pots. So.
Okay, so you had germinated seeds not in the mix original.
No.
Okay, the treatment mix. Okay, that's good to know. What I just wanted to sort of you to consider is oats is a source of carbon, okay, because it's a waste product and it's, you know, mostly carbon, I'm sure. Maybe there's a little bit of protein. That'd be interesting to know. But what I was thinking was that maybe oats would have the effect of a nitrogen drawdown in the potting mix. So I was really surprised to see that adding nitrogen didn't actually help, you know, with using that carbon in the soil. So I'm not sure. Just have a read about what nitrogen drawdown is, and maybe that could help interpret results. But yeah, it's interesting. Thanks very much.
Thank you, Alida. Any more questions? Oh, John's got a question.
So sunflowers are often grown for their seed yield. So did you think about assessing seed yield in your experiment, or will you assess that?
No.
No.
No. No.
Yeah, thank you, John.
Sorry,
Thank
what
you,
was the
John.
question?
The question is, sunflower is usually assessed for seed yield. The answer was seed yield was not assessed. I think the time limitations on the project would make that exceptionally difficult. Any more questions? I have a quick one. My soil, my inorganic chemistry is very poor. I understand that you use sandy soil. Is that correct? Please correct me, someone. Sandy soils are acidic in nature, aren't they? Or maybe I'm mistaken.
Yeah.
Basically. So wouldn't it be preferable, since the ash increases acidity? Wouldn't it be preferable to use ash in alkali soil to neutralize the alkalinity of such soils? Especially if you want to add minerals, because acidic soils are well known for washing out. Because of the acidity, the minerals wash out of the soil when we irrigate. Just a thought, just think about that. All right, thank you very much. If there are no further questions, everybody, please round of applause for Manisha. Thank you so much.
Okay.
Thank you. Yes, thank you. All right, we are 5 minutes ahead of time. May I propose everyone get up, shake it up, go to the loo, or whatever you need to do. I'll give you 4 minutes to do that, and we shall reconvene. We'll keep the recording going, and we'll be back in 4 minutes. Yes, so everybody, please, in the room, get up, shake it up. Where you are, if you're sitting, please get up. All right. Maybe I can stop the recording. I don't know how to do that. We'll just leave it. I think we'll just leave it and yeah. All right.
Yes, you have to throw it. Yeah, thank you very much.
No, I didn't do that. But I have it there enough.
Thank you all. For those of you that are still with us, really appreciate your help. And yeah, please keep marking. Sorry.
Next presentation.
No, not yet. Just for call.
I made few changes in slide.
Yeah.
Was-
It's in the same USB?
Yep.
Okay, leave it with me. Thank you. That's Manisha. So Arun, do you mind?
The other one.
Oh, just save this one and I'll be with you in a moment. So everyone, I'll be with you in a moment, just saving this. Okay, so where is your I'll just close this. We don't need this. So where is your presentation? This one?
Yep.
You are number 6. Okay, we're there. Alrighty, and so we go back to the we start sharing and share, and then we go full screen. Yep. Okay, so I'd like to introduce you to Mr. Arjun Gurung, his supervisor in Ramandeep Singh, and he's going to talk to us about improving post-harvest fruit quality of sweet cherries. Thank you. Please proceed.
Hello, good morning, everyone. Thank you for joining us here today. And I'm here to welcome you to
Stand in front of the camera here.
Yep.
There's a camera. So stand here and talk to the camera.
So I'm here to present my presentation on improving the post-harvest fruit quality of sweet cherries. So let's dive into the presentation. So cherry is not only a beautiful and nutritious food, but it also has a huge economic impact in the world. So talking about the global perspective of cherry, the worldwide trade of cherries is valued at around 2.4 billion US dollar. This data simply highlights how important the cherries are among the growers in the world, and its economic and its market demand in the world as well. So let's talk about the cherry production in Australia. The diverse climatic condition of the Australia, the diverse climatic condition of the Australia provides a very suitable provides a suitable condition for growing different kinds of cherries in different parts of the region, like New South Wales, Victoria, Tasmania, and southern and Western Australia. So Tasmania is one of the main producer, main producer of cherries in the Australia. Talking about Tasmania, simply overall in Australia, it produces around 17,400 tons of cherries, which is worth around $226.5 million US. And talking about Tasmania, it contributes around 1/4 of total production with an impressive 58% of the total export. Total export of the total export from the country. So this highlights how important is it to the grower of Tasmania for the cherry. So it is one of the it helps in the agriculture overall agricultural growth of the Australia. However, no industries is devoid of any kinds of problems. So this cherry industry also faces major issues, and some of them are, one of them is it has very low shelf life, which is just about 1 to 2 weeks. And this will affect the marketability and this marketability of the product in the international market. Similarly, another one is the splitting or the cracking, which generally occurs due after heavy rains or something like that due to climatic conditions. And the other one is the production of the softer fruits, which will be difficult for the producer to handle it, to handle its quality in the future days, because it will be difficult to handle the product if it's very soft, and marketability and the preference of the customers is also compromised. So what are the cherries industry top priorities is to meet the export demand of the market demands. And for this, the key attributes are like larger size fruit, firm and crunchy fruit, bright colored, sweet and tasty fruits. And so these criteria or these characteristics should be maintained to compete against other cherry producer around the world. So these are the major priority for the cherry industries in Australia. So what are my research questions? My research question is how the effect of the infill application of gibberellic acids or the GA3 will have a effect on the post-harvest quality of the fruit, and what are the different treatments we can apply after its harvest and what are its effect. So the aims is to improve the quality of the cherries while in the field pre-harvest, as well as to improve its quality after the post-harvest as well. So these are the few chemicals let's say the treatment we used, and these are simply the functions of what this treatment does to the fruits. So let's talk about the materials and method. These are the experimental details we did for this research. We conducted 2 trials, and trial 1, it includes 2 factorial things. It has like field treatment of control and the control trees and the double application of gibberellic acid. So the fruits were harvested accordingly to separately, and these were, after the harvest, these were treated with the post-harvest treatments like ascorbic, oxalic, salicylic, each of 1 micromole. And similar process was also conducted for GA3. The fruits were harvested separately, and then we did the same procedure. And for the trial 2, we did like we harvested the fruits from the control trees from the protected or rain cover areas, and for the GA3 we harvested from the conventional cropping system. So what we did is, after this harvest, we applied treated with melatonin of 0.5 micromole concentrations, and same was done. After the treatment, they were dried in a separate in a cold store and labeled properly and stored in a cold storage of about 1 degree Celsius for about 4 weeks, and the experiment and the different parameters were measured exactly after 4 weeks. So what are the results that we obtained from the this research? So this we measured the fruit firmness of the cherry, and here you can and similarly the data were analyzed using Excel and SPSS, and all the ANOVA assumptions were tested, and they all made the ANOVA assumptions. So from this result, here we can see that there is a significant difference between the fruits from the GA3 treated trees and from the control trees. You can see that fruits treated with the GA3 has higher firmness, and all the treatments had higher firmness, whereas fruits from the control trees had lower firmness. There are not much significant difference within the treatments. We can say that obviously GA3 treated trees had better fruit firmness as compared to the control. And talking about the flesh firmness, you can, as seen previously, there is a significant difference between GA3 treated and the control one. So you can see the flesh firmness of the fruits treated with GA3 had higher flesh firmness as compared to the control trees, but there were no any significant differences between the post-harvest treatments. And talking about the stem pole force, it's also the same, is as per the what we expected. So you can obviously see the GA3 treated fruits had higher stem pole force as compared to the control ones. So these are well according to the what we had expected. And talking about the cracking, it's quite contradictory. You can see that the GA3 treated fruits had higher percent incidence of cracking as compared to the control one. It's quite contradictory to the results from the different papers. However, you can see that the controls from both the control treatment from both the trees like control and GA3, they had higher incidence of cracking. So obviously the treatments are doing their work in order to minimize the incidence of cracking. So the trial 2, we also measured the same parameters, studied the same parameters of fruit firmness, and you can see that melatonin obviously has has good advantage over the control one, and obviously the gibberellic acid is way is significantly different than the control one. So there is quite this is quite a good results. And the flesh firmness is also the same. Melatonin has the advantage over the control one, and so on. And the stem pole force, it's the same as what we had expected. The chemical is doing quite well, and you can obviously see from the graph over there that the GA3 treated trees are highly superior, and they have good advantage over the fruits in the in maintaining the quality. So, however, in here the cracking is pretty much higher in the control trees as compared to GA3. So it was what we expected, and the control had the higher cracking as compared to the treatments, post-harvest treatment. So these are in correlation with the fact that the treatments like we used, the post-harvest treatment like we use salicylic, ascorbic, and oxalic, they are all antioxidants. So they are the keys molecule to activate the defenses against any kind of stresses for the fruit. And so these are all in correlation with the other findings. So what we can conclude is that the obviously we can say that the gibberellic acid treated fruits were highly superior as compared to the that of the untreated ones. And however, the results have shown the treatments, the post-harvest treatment has shown some differences in maintaining the quality or the maintaining the fruit quality. I'm more excited or more interested in doing further more research in how we can how the a bit higher concentration of these treatments can affect the fruit quality for the control trees. So we need a comprehensive research to carry out to substantiate the above results. So, and lastly, and I would like to thank my supervisor, Ramandeep Singh Sidhu, for his continuous support and guidance, and Adele, Caroline, and the grower, and my colleagues and my family for their unconditional support. Thank you.
Thank you. I'll stop sharing so that we can see everyone. Certainly, thanks should be to Caroline and Ramandeep, not myself, but thank you anyway. Appreciate that. So I think I'll kick off by asking, what's the difference between fruit and flesh firmness? You mentioned fruit firmness and flesh.
The fruit firmness is without peeling off the fruits.
Yeah,
fruit
without peeling off the skin.
Yeah, skin of the fruit, and the flesh firmness is after peeling the skin of the fruit.
Right.
Yeah.
And since John gets 2 questions, I think I'll get 2 questions as well. In malting and brewing, gibberellic acid is sometimes considered a no-no.
Yeah, it's the high application of gibberellic acid is not benefit for the fruit as well.
Yeah.
So it is quite contradictory.
So how does the consumer perceive the use of gibberellic acid in cherries? Is it accepted by the consumer? Or
yeah,
not?
it is accepted by the consumer.
Accepted.
Yeah.
All right, good. There's a hand up by Alida. Alida, please go ahead.
Arun, sorry, yeah. Oh, what have I done? Yep. Hi, Arun. Really interested in your results. You have a lot of results. So well done with doing a lot of work. I think maybe it'd be interesting to see some of the analysis in more detail when you're not under so much pressure with time. But I wasn't quite clear whether you felt from the results that the cover, the cherries that were under cover were better, were not as good as the ones not under cover.
Yeah, it's because from cover we only harvested the from the control trees. So the idea was to harvest the GA treated trees from the cover as well, but there are some variables that we can't control. So because of some unfortunate miscommunications.
Okay, sorry. I thought you must have got your plants, your cherries early. Okay, ignore that question. I didn't realize that. Look, I think it would be nice to know a bit more about why you chose melatonin. So I did a little Google search because for me, I'm into cherry research, but I'd never heard that you could use melatonin to as a post-harvest treatment.
Yep.
And it was really interesting to read that melatonin is actually naturally present as an antioxidant in sweet cherries naturally. So you're actually just boosting the melatonin in the cherries already. And apparently there's some promising results. So that's really good to know. And that's exciting research. Thank you.
Thank you, Alida. Anybody else, please? Was who? Bianca. Bianca, please go ahead.
Hi, sorry, I did come in a little bit late, so I might have missed some key things, but just a sort of general question. I sort of wanted to challenge that question around, is gibberellic acid accepted? It's quite interesting at the Launceston markets, there is a cherry producer who talks about how bad hormones are for cherries, and now he grows his cherries without the hormones, that are, you know, kind of goes off on a rant about that. And I've sort of said, well, from an agronomic perspective, it does actually make sense to apply these sorts of products, even because they are naturally occurring hormones, which he didn't really like me saying. But yeah, just curious, when the word hormone gets used, people get scared. So is there some sort of marketing, or like, do you have to be careful about that?
I don't think it has the higher dose of GA3 application is obviously not good for the fruit quality. I don't know about what it does on the health perspective, but in terms of the fruit quality and other parameters, it does not have the higher doses is certainly not recommended. So, however, we only applied like 50 ppm of double applications, which is relatively in between higher and lower, so it's in the middle.
Okay, so it's really about the dose determines the poison. Yeah.
Yeah. Yes.
If I may step in, it's about consumer perception as well. No matter what the dosage is, usage of hormones on many occasions is on the nose. Are there any more questions for Arun, please? Well, Arun, thank you so much for your time. Really appreciate the talk. Seriously, it was really interesting. Thank you.
Can I take the...
Yeah, I'll give it to you in a moment. Thank you, Sibi. May I invite Miss Rupa Ghimire. Her supervisor is Tori Clark, and she is here to talk to us about effects of RNA carrier clay on grapevine physiology and disease susceptibility. So I'll just oh. So where are you? There's Rupa. I'll just close the extra ones. Rupa. Okay. All right, let's share screen. Nope. So share screen. There's Rupa. Are you nice and relaxed? Come on, breathe, come on, breathe. You can do this. You can do this. Just come and stand here so everybody can see you. Front of the camera here, and away you go.
Hi to everyone. And my topic is effective RNA carrier clay and grapevine physiology and disease susceptibility, and my advisor Tori Clark. So before going to the next slide, I'll just give a little bit overview of my research. What I'm gonna do is about the botrytis disease and related about the bioclay and what's the effects of bioclay in plant physiology as well as the botrytis growth and some conclusion. Okay, then botrytis, this is caused by the fungus called the botrytis cinerea, and it is second most phytopathogenic fungus, and it's commonly called like VBR, gray mold, and bunch rot. And this is affecting the grapes and reducing the crop yield and the quality, and then it also reduces the quality of the wine. And in the wine industry, the processing cost is like really high because of this botrytis problem, and also increase the production cost by the 24%. And okay, what is happening to control this? Then people are using a lot of biological control, chemicals, and lots of things. And we all know that we are in the era of the chemical revolution. That means we are, minimizing the use of chemicals. So we are using the fungicide to control this. But with the grapes, after 80% cap full, you are not allowed to use the fungicides. So it's like, okay, what next, and what gonna we do to control this disease? Because like in wine industry, and it's really the important thing in Australia and all over the world too. So I want to introduce to this technology, which was called BioClay technology, and this technology is working well with the help of Sustainable Crop Protection ARC Hub, and it's like sustainable, biodegradable, and non-genetically modified. Okay, I'll explain a little bit about this technology. Is like they are spraying dsRNA in the leaf, and dsRNA in the leaf, and it will interference the the RNA of that targeted gene, so that there will be no fungal growth. And the hero of my my work, that is LDS, which is called Lear double hydroxide. When you use that as a carrier in that system, then it prolong the reduce the fungal growth. There are some example like in tomato, in tomato, in fruit leaves, there is like it increases the like 10 days for the disease incidence like lower, and in fruits also it for like 10 to 30 days. So this LDH, it can have like really good effect on putting that dsRNA for the long time, so that there will no fungal growth further. And okay, then I was talking about lots about LDH. Then in my thesis, I want to see, okay, what happened in the leaf physiology as well as the disease aspect. So I'm looking like the two aspect. One, will spraying LDH clay onto the grapevine leaves have detrimental effects? And my next question will be like, does LDH clay reduce the disease incident and the severity? And regarding this, I told you that before, clay is evolving, and then there's not that much research going around the clay itself, because as I mentioned before, that LDH clay act as a carrier and hold that dsRNA for the long time. Then okay, we want to see further, does it have like some effects on those things? Okay, then what we did with the methodology, then this experiment was conducted in the Brunei in glass house, where like 40, we call it, grapevines cutting were propagated, and there were like 6 biological replicas, and here are some treatments we have been using in that experiment. There was water, switch, and the 0.1, 5, 10, 15. These are my different concentration of the LDH, and we want to see that, okay, applying these treatment, they will have some effects or not. Okay, before that, we just choose like one leaf, like a young, which have the laminar length of 62 to 63 mm, and then we just level it. And before that, we also measure the chlorophyll fluorescence through the multispec. I think my friend Sudan also explained a little bit more about the multispec in previous presentation, so I'm adding on that too. So, and then we just observe, we spray that on the leaf, on the both side, and then see the result like till 2 weeks. And now we measure the chlorophyll fluorescence using the multispec. So I've been talking about the chlorophyll fluorescence since long. Then okay, like normally we all know that photosynthesis phenomena just convert light energy to chemical energy, so that you can have like plant can have proper growth and everything. So there are several factors that affect the photosynthesis. It can be light, it can be leaf, water, everything. And I'm focusing most on the stress, because my thesis is related with that. That does, if you apply clay, does the plant have some stress or not? So from that picture also, you can see that when there is light is coming, then plant cannot utilize all the energy. It will go either in the form of heat and fluorescence, and some will utilize the photosynthesis. But you can see in the next picture that when you apply, when there is same enough light is coming, but when the plant is stressed, then you can see that more of heat and fluorescence is going out, and you can see that green arrow becoming a little smaller, and that means less photosynthesis activity have been done. So we can these red arrow CF, that is measured by the multispec, the next important tool of my research. Okay, then the next question, can we capture that chlorophyll fluorescence? Yes, with the multispec, because it's really easy, low cost. It just take like 50 seconds. You can, it was so easy that you can upload all your data into the tab and then calculate it by yourself. And then it can identify the biotech as well as all the stresses. Even I was so happy that knowing, it will take you like when you take the time, even in location, and it measures lots of parameters required for the photosynthesis. And then like some of the most important parameter it was measuring were like phi2, phi-NO, phi-NPQ, and the LEF. These are the parameter which will show, okay, because these give like details of those numbers so that we can calculate and see that, okay, does the plant have some stress because of applying those treatments or not. So I have done like phi2 analysis, phi-NO, phi-NPQ, lots of things in my research, but now I'm only focusing on the LEF, which is linear electron flow, and it just assays the utilization of the ATP and NADPH, and then it's also the important parameter of the photosynthesis. So I do have some research. I do have some result with this, and I have in lots of literature review there was given that when LEF increases, the photosynthesis also increases. So and when it decreases, then it also decreases the efficiency of the photosynthesis. Always also shows, okay, plants is showing some stress. So for that, I'm going to show you some like what happened. Like when you apply, these are my day 0, 1, 2, and 6, and the LD, there are some colorful LDH concentration, and then LEF is on that side, and you can see that when high LDH concentration have reduced the LEF in grape leaves, you can see that those blue and purple are like my highest concentration, and you can see there was like reducing of the photosynthetic activity. That means plants show something. So you can see in the first day and second day and third and the sixth day, we can see like some changes in that. So I do that with the I do the analysis with the R studio, and then get to know that there are like some significant difference with the mostly like with control. You can see there was significant differences with the LDH concentration 10 and 15. So that means when you apply LDH on that plant, then there is change in the photosynthesis thing, and further then I'll little bit focus mainly on like two factors here. You can see like when I take like zero control and the 10 concentration, you can see there is like decreasing of LDH, decreasing of decreasing at the 0 points, and then again you can see when it go to like 1 and second and further, that's increasing. That means we can see that plant get a stress when you apply that treatment, but after some time it will recover. It will take at the same position, so it will get recovered after some time. So these were observed in my experiment thing, and then further analysis, because I have also done some image analysis to image J also, and where we can see that when you apply like only water on the because we spray like water and all those thing, and when you apply the water, then you can see the in like 0, 2nd, and 6th days, you can see like there is the plant shows in water conditions some discoloration, and maybe because of aging or something. But when you look at the picture where it's applied at the high concentration of LDH, you can see some discoloration as well as some lesion size. Maybe that may be the cause of the phytotoxicity. I cannot predict now because I still need to analyze like more pictures from that one. And so these are my answer of the first question, where I first research question where I was targeting only the leaf physiology thing. And my next part was like, does that clay affect the botrytis growth or anything like that? So again, in the Bruni lab, we were doing like preparation of the botrytis cyanida and all the treatment. We are preparing that, and for this experiment, what I did, I also did like plate assay, and then I was following the droplet assay and the spray assay. But for now, I'm focusing on the droplet assay here. That what we did, like we normally took the grapes, arrange it, use all the ethanol, everything, cleanse it, and then do proper, process in the laminar flow. We put spores as well as treatments, and then we scored the grape based on those pictures. And then what we get after that, then we can see that when, because we were using like, you can see the below picture, like lower concentration, there is high incidence of disease. When you increase the higher concentration, like at LDH high, you can see there is less incidence of disease. And then those upper were my control, where I was using mock and the switch and water. Obviously with the spore, switch can control the spores, so that was really good for me. And then with water also, there was with a mock test also, there was no growth of fungus. That means I was very happy with the experiment. And again, I repeat this like twice, and the result was also the similar. Like you can see that after repeating that result, so I can say that when you increase the dose of LDH concentration, then there is like decreasing of disease incidence. And you can see there are like significant difference with high concentration and below 1. So it was like really good to see that thing. And but with a spray that was not similar with this. So that's why I can conclude through all my work that higher concentration of LDH initially reduce the photosynthetic efficiency, but plants adapt some time, and then LDH alone can suppress the botrytis growth. This may provide an additive benefit to dsRNA disease suppression when formulated as biocally, because I already mentioned that droplet assay was doing really well, but maybe with a spray it was not expected. So I think this would be the great to see further. And then because I think still more research need to be done, because there was not that much experiment like going on with the LDH clay. And yeah, that's the thing. And these are my reference. And thank you. I really want to thank each and every person helping me, especially Tori and Akim and the Bruni Glasshouse and whole train. And yeah, that's all. Thank you.
Thank you so much, Rupa. That was really interesting. So many interesting topics today. I'll just take off sharing. Any questions, please, for Rupa? So Rupa, if you stand in front of the camera so they can see you. Yeah. Any hands up? Anyone? Yes, sorry, Shane, please. I wasn't. Yeah.
Because it have been affecting like lots of crops like tomato, and then because like major commercial plants, and grapes is like really a commercial here in Australia too. So
that's just that high concentrations of the LDH may have get mentioned. Is there a spot where you could apply a lower concentration where it could be useful without?
Actually, like normal recommendation dose were the 0.1, and we just want to see that does plant get stretched or not. So we have like level from low to high, and with low, I think there was not that much significant differences. So I was like highlighting more that more have, like when there was like high concentration, there is like stretch in the plant. With low, I think, yeah, that's why I was like, okay, yeah.
Any more?
Yes. Sorry,
yes,
Adele.
Oh, yes, please.
I'm not sure. I didn't hear quite Shane's questions. I think
Shane.
No, it's okay. I think I might have got it, but just in case if it's a repeat, please just let me know. Good job there, Rupa. I'm just wondering to know, you had a range of concentrations of the clay technology. Just in terms of previous literature, how do you sort of relate that to the concentrations in previous literature? So is 15 a very high concentration, or is that, relative to other literature, do you feel that that the results are comparable with other studies? That was basically my question.
Actually, when I was searching with the clay thing, there was not that much research going on. Like, because only because it's related with the dsRNA and that terminology at the bioclay. So that's why, and the normal recommended was 0.1. That's why we want to see with the dilution theory, maybe does it work? Does it have some effect with lower to higher? And we can see that with when you have like higher concentration, that the plant get like more stress.
Yeah. Okay. So in your I know you've done maybe the lab has overall done some studies in this area. Okay. Thanks, Tori. She's written down in chat, current formulation is 1 gram per liter. So 15 is huge. Okay. That's good to know. Thank you.
All right. Well, any more questions, please? All right. No one online, no one here. Thank you so much for your time.
Thank you so much, everyone.
Thank you. Right. So the next speaker, I'd like to call Fatimah Umatiya, please, up here. She is a student of Dr. Saifullah, and I don't have your title. So
application of cherry antioxidant in the
would you like to say your title, please?
That's application of cherry powder as a natural antioxidant in ready-to-eat meat processing. That's my topic.
Right. So I've uploaded you send it
Yeah,
to me?
this is a bit revised.
Oh, a
one.
bit revised again. All right, lovely. All right. So we'll do that. We'll do that. Okay. So let me close down your
Mhm. The one that's open.
we'll close this one as well. And all right, so let's go to file.
Sorry about that.
It's all good. We'll sort this out shortly. So yeah.
Yes.
And which one is it?
Fatima? Yeah. Good.
Fatima. And we'll just where's my mouse? Oh, doesn't matter. There's Fatima. We'll share Zoom and share. There it goes. And then we will. Why can't I see the bottom of this? Wait a minute. Yep.
Yep.
And boom, there it is. And away you go.
Hello, everyone. My name is Fatima, and today I'll be presenting my project about, where's the enter?
Yeah.
About application of cherry powder as natural antioxidant in ready-to-eat meat processing. So I'm doing the more food science part of the Masters of Agriculture and Food Science. So first, we all are very aware of the growing demand of meat products in today's fast-paced life, where according to a report from DAF, there was an increase of consumption of meat product of over 58% from 1998 to 2018. So this depicts the increasing demand of meat product. Now, I'm very sure everyone who eats meat at least must have experienced that this bright red color of meat turns to this gray dull color. Alongside the other undesirable characteristics which everyone might have experienced is of odor, slimy texture, and we wonder that this is the meat spoilage. But what causes the meat spoilage? So basically, meat spoilage is caused by three mechanisms. The first one being microbial spoilage, which according to Dave and Gelly, they explain that meat is an excellent source of nutrition for microorganisms and bacteria to grow on, which is also dependent on several other factors like temperature, pH, water activity, etc. And it usually results in the undesirable characteristics like we saw in the earlier slide. The other cause of meat spoilage is autolytic enzymatic spoilage, which in normal terms is self-deterioration of meat muscle by the enzymes present in meat muscle, and it forms a basis for microbial spoilage. The third cause of spoilage is oxidation. So oxidation is basically the natural chemical processes occurring in meat itself, where the lipids and proteins oxidize in the presence of oxygen, which usually results in loss of sensory qualities and similar to the microbial spoilage. And lipid oxidation is again basically like primarily classified into three types, which is auto-oxidation, photo-oxidation, and I don't want to go deep into it, but basically it all results in the loss of sensory qualities of meat. Now, how can we stop oxidation? So literature suggests the use of antioxidants, which are basically compounds that scavenge on the oxidation products. So oxidation results in the release of free radicals and reactive oxygen species. And these antioxidants are compound that scavenge on this oxidation product, resulting in inhibition of oxidation and then preserving the freshness of meat and the quality of meat. So antioxidants are basically classified into two types: synthetic antioxidant and natural antioxidant. Synthetic, like the term is, are chemical compounds which are synthetically prepared. But like in recent years, there has been a growing concern over safety of those. Commonly used antioxidants are BHT, BHA, propyl gallate, which have long-term health implication on humans' body. And some studies even prove that they have carcinogenic effect on human body in long term. Natural antioxidants. Natural antioxidants are basically compounds derived from natural sources like fruits, vegetables, herbs, spices, which are rich in polyphenolic and bioactive compounds. And they work in the same way as synthetic antioxidant compound. One of the fruit known for its rich antioxidant potential is cherry. So there are several studies. Due to the slide restriction, I could just list 3 which were related to my work. But there are several studies which have proved and investigated the potential of cherry as natural antioxidant in meat. Most of the literature is focused on raw meat at refrigerated temperature, and this research gap forms the basis for our research, where we thought that why not use cherry antioxidant in shelf-stable meat. Also, we use cherry powder, which was sourced from second-grade cherries, which is basically cosmetically damaged cherries not suitable for sale, usually goes to fruit waste. So the advantages of using second-grade cherries is it becomes a sustainable approach and a cheaper alternative if proven that its effectiveness in shelf-stable meat forms a sustainable approach. And also because lately in recent years, consumer demand for meat products with natural ingredients have increased. So it meets the consumer demand for natural products as well. These lead us to our research aims and objectives, where we aim to achieve long shelf-stable, ready-to-eat meat product by the following 3 objectives. The very first, applying natural antioxidant, that's cherry natural antioxidant in shelf-stable meat processing. The second one is utilizing second-grade cherry powder or second-grade cherries byproduct in enhancing the shelf life of ready-to-eat meat. And then and also we need to know that, okay, does cherry powder remain stable after processing or not? And that is our third objective of our research. The specific research questions we aimed in our work were, what is the impact of adding cherry powder on the oxidation levels in meat? The second was, what is the stability? Like, does it remain stable or does it lose its activity after processing? And then also one of the important aspect of shelf life of food product is microbial activity. So does it inhibit, like, does the addition of cherry powder inhibit the microbial growth or not during storage? The experiment was designed in a way where there were 6 different treatments. The first one being negative control. The second one was being a positive control, where a standard concentration of BHT, one of the synthetic antioxidant, was added. And the other 4 treatments were 4 different levels of cherry powder, ranging from 1% to 7%. We analyzed the samples on 4 days. That's day 0, day 1, day 4, 7. And each treatment had 3 replicates. We prepared our patties, where we specifically focused on beef meat. So we took beef mince, we added cherry powder for our cherry treatments, BHT for our positive control, and for negative control, nothing. And then we took 10 gram, we weighed 10-gram patties, vacuum packed it, because vacuum packaging is one of the effective way of inhibiting oxidation as well, because there is no oxygen in there. And then according to some preliminary test, the patties were heated for 70 degrees Celsius for 10 minutes. This method resembles a sous vide cooking method, which is defined as cooking for longer time at lower temperature, which preserves meat's quality and freshness and taste, according to literature. Because our sample size was very small, that like the sample weight was 10 grams, the preliminary results reveal that, okay, the core temperature is the same as like this 10 minutes and 70 degrees was enough to cook 10 grams. And those were stored at room temperature, which in our project was 22 degrees Celsius, and then analyzed on day 0, 1, 4, 7 for the following parameters. The first parameter was, of course, lipid oxidation, which was analyzed using T-bars analysis, which looks at the specific oxidation products and quantifies them as well. The second parameter we looked at was the antioxidant stability. The first one answers the first research question of the oxidation levels. The second one answers the second research question, that is the antioxidant stability using different antioxidant assays. The third one is the microbial growth using total viable count under anaerobic condition. And the fourth one was the physiochemical parameters, which was pH and color. However, for the rest of the talk, I'll just be speaking about parameter 1, 3, and 4, because the data collection for the parameter 2, that's antioxidant stability, was completed yesterday. We are still yet to analyze the data for that. And the data analysis would be done for that using SPSS and Excel, using one-way ANOVA factorial design. Now let's look over the results. Yep. So the first result is about the oxidation, which was a main focused main aim. That does cherry powder or does cherry antioxidant influence meat oxidation level? Now let's just have a background information about what we looked at. So MDA is secondary oxidation compound, which is melandoaldehyde. It forms a pink colored complex with TBA. That's what we reacted it with. And then the increasing intensity of the pink color depicts the increasing level of oxidation in meat products. So what we found out that there was an increasing trend in the oxidation in all treatments between like with the increasing storage time. However, the assumption was that the highest concentration of cherry powder should give us lower oxidation. That means that, okay, cherry powder is working, and that came out as expected, where on day 4, okay, does it say 1, 2, 3, 4?
Yes, it does.
Oh, okay. So it is 0, 1, 4, 7. I'm so sorry. It was just overlooked.
Do you want me to move it?
No, that's all right. That's okay. So it's the time below is, the first is 0 day, the second is first day, the third is 4th day, and the fourth is 7th day. I'm not very sure as to why that was just overlooked, but okay, it's 0, 1, 4, 7th day. So the last graph is for the last day of analysis, that was for 7th day, and the first one is for 0 day. On each day, the highest concentration of cherry powder showed the lower oxidation level, and that means that our cherry powder is working in inhibiting oxidation. The second parameter so these were the key findings of our results for oxidation analysis. The second parameters was color, which we looked at the difference in color before heating and after heating, and then between treatments as well. So before heating, this was the color of all different treatments, where the cherry powder with 7% cherry powder meat samples has the darkest color. However, after heating, there wasn't any visual difference found in the color of the samples, but there was oxidation happening in there as seen in the previous slide. In regards to the pH, the pH on day 0 was lower for the highest concentration of cherry powder. It might be due to the higher content of ascorbic acid, but that's vitamin C in cherry powder, and that might be the reason that with increasing concentration, the pH decreases. And over the day, like over the storage, over the storage period, there was a decline in the trend observed that the pH decreased for all the treatments. And one of the explanation for this is because there was microbial growth observed, and bacteria can break down and release like different acids, which can result in decrease in the pH in all the treatments. Then next result is about the microbial growth. This was one of the most important results we observed. We haven't analyzed the data like statistically in numbers, but owing to the graph, we can see that cherry powder was ineffective in inhibiting the microbial growth on all days. And this formed a significant shortcoming for our result, where we observed that there was lack of inhibition at 22 degrees Celsius for our treatments. And this can be explained using literature that 22 degree, like temperature about 20 degrees is like an optimum temperature for growth of anaerobic bacteria. And this forms a basis for a future research opportunity where if there is a further investigation required for cherry powder's antimicrobial activity at lower temperature, and if that is further optimized, it can result in optimization of preservation techniques for using and harnessing the antioxidant potential and antimicrobial potential of cherry powder in enhancing the shelf life of meat products. Done?
Okay. Okay.
Do I still have time?
Well, you finish, but
Okay.
you've gone over.
Yeah, no, I've completed the conclusion in the last part, but yeah, that's that. If proven in further research, if cherry powder's antimicrobial potential is proven at lower temperature, it can form a basis for a suitable alternative for synthetic antioxidants. That's done. And then these are the references and questions. Yeah. And also,
Sorry,
yeah,
hold on.
sorry, I got disoriented with the time. That I would also like to thank my supervisors, specifically, who were very supportive with me in my research, which were Mohammed Saifullah, Naomi, Roger, Jay, and also the staff, which they were really helpful with me understanding different things in the lab.
Thank you, Fatima.
Yeah.
Thank you very much. Thank
Yeah.
you. John, please.
So will the addition of the cherry powder, will it affect the way that we taste?
So we haven't tasted the meat because we first wanted to know, is it safe over the time? Like we just wanted to know about the oxidation and about the microbial growth, like for this. But if it is proven, then we might go into sensory analysis later on, or when the research is being further carried on.
Do you think it would give it like a cherry kind of flavor?
There wasn't any difference in the smell because I was the one who smelled it, but that depends on when it's tasted. Yeah, but maybe lower concentration might not affect the taste at all if it's coupled with different other ingredients. Yeah, it might be. Yeah, other questions, please, if there are any. Yes.
Nathan.
Yeah. I was just wondering if you took a pre-cooking sample of the mince after you mixed it together.
Mm-hmm.
Do you know if there's any difference, like in terms of the cooking process, like with and without the cherry powder? Is that going to change the way it cooks or anything like that? I don't know. It was just, I didn't see a pre-treatment sample.
pre-treatment sample. Oh no, the pictures in the PPT where it was before heating, that was the pre-treated
Yeah.
sample, and the after heating was the after treated sample.
Did you measure any of those other parameters though,
So
like the oxidative stuff?
I did not measure the oxidation, but we did measure the antioxidant potential, but the data, as I said, is still to be analyzed, and
Right.
we're still waiting for the analysis.
Chen.
Yeah.
My question has been answered by Nancy, because I was about to ask, have you measured the content of the antioxidant? You said you measured the potential, but
Yeah.
you did the analysis. Yeah.
So the different assays which were used for measuring the antioxidant potential are also like they also quantify the content of antioxidant present in the samples. So we have the data for before heating and after heating and over the storage period as well. But as I said, it's still yet to be analyzed.
Just have a question, maybe, so stupid. Because, in the cherries, a lot of sugar. Is sugar going to be an issue affecting your resource? Or do we need to consider like the involvement of sugar in cherry?
Maybe, so we were expecting to see less microbial growth with the increasing concentrations of cherry powder, but then it did not affect the growth. So maybe like the sugar content, as you said, might be one of the factors affecting the microbial growth, but we did not go into the sugar content of cherry powder at all.
Okay, thank you.
Just from the perspective of food science, Chen, the powder would reduce water activity. And I also expect when the burgers are cooked, it would enhance the yield, meaning reduced cooking loss, reduced loss of fat and moisture during the cooking of the burger. Yes. Any more questions? This is excellent. This has been an excellent talk. Any more questions? Excellent, Fatima, you've done exceptionally well. Very impressive. Thank you so much.
And I'm so sorry about going over the time.
No, not at all. Not at all. Not at all. Okay, last but not least, Pratima Karki. Miss Pratima. Pratima, sorry, apologies. Pratima, my old eyes. Pratima Karki, please come over. Pratima is with Dr. Nathan Tivendale. Apologies, Nathan. Apologies. And so she's going to talk to us about hormonal control of pyrethrum growth and development. Yeah. Take it away. Thank you so much. Stand in front of the camera so people can see you.
Can I just have a quick look on my presentation?
Absolutely. Excuse me. You're number 9. So that's it. Do you want me to flick through the slides?
Oh, no.
No, you're happy? Yeah. Okay. Fatima's happy. Let me share the screen with everyone. Okay. Share. And then full screen. Away you go. Thank you.
Thank you, Dana. Hi, everyone. I'm really delighted to present my research on phytohormonal control of pyrethrum growth and development. This is Pratima Karki, and I worked under the supervision of Dr. Nathan Tivendale. So yeah, I really appreciate your presence today, as we're going to speak in the idea of how to enhance the pyrethrum production through the use of phytohormones. So moving forward, we will actually cover these key areas. We'll talk about the introduction and objectives of our research, the methodology, whatever we did, the results and discussion, and conclude our research. So let's start with what actually is pyrethrum. Pyrethrum, scientifically known as Tanacetum senalifolium, is actually a perennial herb that exactly looks like a daisy flower and belongs to the family Asteraceae. The first ever cultivation of pyrethrum was recorded in around 1840s near the Dalmatian coast. Talking about the major producers of pyrethrum, they are East Africa, Tasmania, China, and Papua New Guinea. So talking about the specific growing requirement, these plants require moderate watering, well-draining soil, temperature requirement of 15 to 25 degrees Celsius, and vernalization temperature of less than 12 degrees Celsius night. So why do we think pyrethrum is so important, even though it exactly looks like any of the daisy flowers? It's because of its insecticidal property. Pyrethrum contains this unique compound called pyrethrin in its oil gland of the seeds, which is actually used as making insecticide. Talking about pyrethrin content, pyrethrin consists of 6 insecticidal components: pyrethrin 1, pyrethrin 2, cynidin 1, cyphenin 2, jasmonine 1, and jasmonine 2. Being pyrethrin 1 and pyrethrin 2 dominant with the 73% of concentration in the oil glands. So yes, when the flowers are dry, they are crushed and grounded to form powders, and then later on they are used to make spray to kill, or let's say repel, the insects. So moving on, so yes, the main reason why we are doing this research is it might be surprising that even though Tasmania is one of the major producers, but BRA, Botanical Resources Australia, which is actually Tasmanian-owned company, produces more than 60% of pyrethrin insecticide in the whole world. So from this, we can actually conclude how much potential this Tasmania have in producing the pyrethrum. So yes, also Tasmania is actually blessed with the climate. So most of the pyrethrum production is done in high latitude and low elevations of less than 300 meters, and the temperature of less than 12 degrees Celsius, night temperature, that is required for the vernalization. Every standards Tasmania meets it. So of course, if we talk about production efficiency, so yes, given an example of Kenya, which is actually one of the major pyrethrum producing country, the whole life cycle of pyrethrum takes 3 years to get the first harvest. Likewise, in the Tasmanian case, pyrethrum just takes 18 months of life cycle, and the first harvest is done within the if the first plantation is done in the spring and the plants goes on growing for a whole year, then the first harvest could be get in the following summer. It's quite exciting. And also the agronomical practices that have been using in Tasmania is much more mechanized than what people have been using in Kenya. So of course, with the extended juvenile stage, the plant is susceptible to weed, fungi, and many more insects and pests. So of course, if we actually focus on this specific research on how to improve the production, then we can also mitigate this challenges and overcome with the solution. So yes, you might be wondering, of course, if you talk about any of the plants, then there are many numerous internal and external factors that affects the plant's growth and development. And one of the factors that actually helps internally helps plant to have a vigorous growth and development is phytohormones. Phytohormones are the plant signaling molecules that helps in regulating plants' growth and development. There's 2 types of growth hormones, phytohormones, growth promoter and inhibitors. And overall, we can say that phytohormones actually helps in plants' regulation, growth and development, and respond to the environmental stresses. So yes, we are actually using the phytohormones, but of course we don't have any idea which phytohormones we should be using. Is it auxin, gibberellin, cytokinin, ethylene? Which should we be using? Because our main focus is on increasing production of pyrethrum. So yes, we are actually focusing on phytohormones, and the main reason why we are actually choosing GA over any other phytohormones is because it is evident that GA helps in regulating plant growth regulators, and of course it enhances the flowering and everything. And also we have no talking about the knowledge gap, we have no idea if actually GA is involved in enhancing the pyrethrum production as it enters the over-dormancy period. So yes, we are actually assessing the GA, but yeah, there's there's a line that we are not actually working on external GA application, because it is evident we have different literatures and research that actually say external GA application actually helps in enhancing the plant growth and development. But we are not sure if the endogenous GA in plants that are present in the plants actually affect the growth and development or not. So in this research, we are actually focusing on endogenous GA that is in the pyrethrum as it enters the over-dormancy period. So moving on, yes, this is our study objectives. We will be validating this method to measure the GA content in pyrethrum. And we will later on use that validated method to measure the change in GA content as it enters the winter dormancy period. As talking about our hypothesis, yes, our null hypothesis says that we will eventually not see any of the changes in our endogenous GA as the pyrethrum enters the dormancy period. But our alternative hypothesis says that, yes, we will see some significant effects and as the pyrethrum enters that period. So yes, talking about this methodology is actually just specific to what we did for our validation method. So these are the methods that we used for our validation. So firstly, with the leaves, we took our leaves, we grinded it, we added the cold extraction solution, which is 75% of methanol, 20% water, and 5% of formic acid. Then we added internal standard, centrifuged it in 3000 RPM for 3 minutes. Then we extracted that solution. Then we dried our sample. Then again, we added acidified with HCl and ethyl acetate. We let it settle down and pulled out the ethyl acetate phase. We then again dried our sample, then added 50 microliters of EDC, which is really important process in our whole method. Then we sonicated for 1 minute, then incubated at 40 degrees Celsius overnight. And afterwards, we again dried our samples, yes. And then we re-dissolved 15 microliters of molecular water in our samples, centrifuged it at 13,000 RPM for 5 minutes, and then extracted the solution in the autosample vial, and then ran it in LCMS chromatography. So yes, talking about our results analysis. So yes, as I talk about our method validation, we had 3 parameters that we looked after: linearity, reproducibility, and limit of detection. We did linearity tests just to make sure that the method we are actually using is accurate, reproducibility to make it stable and reliable over the time, and limit of detection to determine if the method is sensitive enough. I really want to apologize because we don't have any exact data and result on reproducibility, but yes, we do have some on linearity and limit of detection. As we move forward to linearity range, so as you can see, our GA1 analyte, GA7, 4, and GA20. It does show a really good fit to our R-square value and doesn't show that much of variations. But if we move on to our GA3 analytes, it doesn't fit to our linear range. So yes, from this, we can actually conclude that whatever the validation method we use, it can actually help us give a good measurement of our GA analyte 1, 7, 4, and 20, but it is not good fit for GA3 because it didn't show a good linear fit to our test range. Moving on, limit of detection for our GA analytes. As you can see in the graph, the GA20, yes, the limit of GA20 is much more higher than any of the GA analytes, which actually implies that the detection of GA20 is really hard when it is actually present in really lower concentration because it is really sensitive. And however, for GA1 and GA4, the limit of detection is less. So that means, as you can see, mass on the column. So even though it is present there in less than 2 nanogram, it is actually detectable, and that's good to hear that. And of course, we had some interesting findings when we did our validation results. So firstly, we actually observed 2 of the different peaks when we did our validation in GA3 analyte and GA4 analytes, and the main reason behind this is derivatization method. Let me just go back to my methodology where I mentioned that 50 microliters of EDC is actually a really important process in our whole method, and that's what it is. So usually derivatization is done to make sure that we actually chemically transform one compound into another compound so that it could be detectable and sensitive and it's stable. But during our validation result, that derivatization somehow created new isomer, which actually interacted with our GA analytes and showed one major and another minor peaks for our GA3 and GA4 analytes. Similarly, change in retention time. Of course, if we're talking about retention time, every GA analytes have its own retention time. If we do a chromatography of noise to signal ratio, then there is exactly at one specific time will GA analyte show its signal. So in our case, when we actually spiked our standard solution, then it showed one retention time. But when we ran our own sample extract, then it showed us huge difference in the retention time. So it can be because of the matrix effect of pyrethrum extract, because we are not sure what kind of compounds are present in the pyrethrum. So it could be that the derivatization just made some other compounds and which actually interfered with the retention time. So yes, this is about our field samples. So just to make you clear, the field samples are taken from Hagley, which is 30 minutes drive from Launceston. So this temperature, transport temperature is actually, we had to get our samples back same day here in TIA. So the temperature transport is actually the transport temperature we carried out just to make sure that there is no any effect of temperature on the GA content change. So we did temperature transport on three ones, one with just the skin, nothing else, one with the dry ice, and another with the liquid nitrogen. And when I did one-way ANOVA with our analytes with the relation of temperature, then unfortunately we didn't see any of the changes in GA concentration. So we can actually conclude that the transportation area, or transportation temperature or the storage temperature doesn't affect the change in the GA content. So moving forward, this is our concentration of GA analytes over the period of time. So there were 4 time points: 0 days, 13 days, 28 days, and 35 days. We took our samples at 4 different time points. And as you can see, the GA1 and GA3, the R square value is really less. It's not even near to the 1. So, and similar goes with our GA4, 20, and 7. So yes, we can actually conclude that there was no any relation with our of GA analytes with the time period. So unfortunately, we had to accept our null hypothesis, which said that we wouldn't see any significant effect of GA concentration over the time period. So lastly, let's conclude our research. So of course, we didn't see any effect on the GA analytes with this storage temperature. And we didn't see any changes on any of our GA analytes over the time period, and no correlation of GA with the time period. So we recommend a further research for a really long time, because the research we took was just of 35 days interval. So it wasn't pretty sure, we weren't pretty sure if that is actually accurate or not. So it is advisable to take the concentration level study on a longer period of time, let's say for a year or like at least 6 months. So it would be visible and it would be clearer to interpret the data. Just thank you so much, everyone, for your patience. And lastly, I would like to acknowledge everyone who directly and indirectly helped me complete my research with their support, motivation, and love. Thank you so much. And yes, I'm open to have any questions if you have. Thank you.
Thank you so much. Thank you.
Thank you.
I'll just stop sharing. Okay, please. So stand in front of the camera and
Yes, any
questions,
questions?
please. Questions. Right. Okay. Well, I'll start.
Yeah, sure.
What led you to think that gibberellic acid would have an effect? I mean, in my line of work, when we look at malting and malting of barley, we want more enzymes to develop, because enzymes are important for breaking down carbohydrates, proteins. When we look at seedless grapes, the berries, we want gibberellic acid because we want a larger berry on the vine, so that we make more money.
Yeah.
What brought you and to this point where you thought, gibberellic acid is the answer? How did you come to that?
Well, it's not the answer we're looking for. because we are not even sure at this point that GA is the only that thing we're after. It's not like we are just looking after the GA. It's just our first step to know that what is that thing that could actually help us. Like, the main focus is our pyrethrum production, of course. We just want our pyrethrum production to increase, and we can somehow be the world leader. So yes, GA is just our first step to know if we could achieve that objectives. But yeah, if we could have a better research and it could go on for a longer time, then yes, we would probably have answer one day.
So what other chemicals, enzymes, hormones?
Yeah, we have a lot of prospects for those things.
A lot.
Yeah.
You don't want to say right now. You don't want to
No.
keep it a secret.
No, it's not like we're keeping it secret, but yeah, as I said, this is our first step. So if we could see any effect on GA, then we could possibly work with the GA. And if doesn't, then we have a lot of prospects. We can go with the enzymes, we can go with the temperature. There's a lot of steps we do have. So yeah,
well,
I think I answered your question, or I
no, you have. Talk to Nathan and see
Yeah.
if we can turn this into a PhD, and maybe I can get involved. Okay.
That's
That's right. That's right. I'm just trying to find job security for myself. That's what I'm trying to do here. Any more questions? Anyone on screen? Anyone here? Students, you have asked no questions whatsoever. Students usually are the smartest because you've got fresh minds, not like our minds with the cells dying. But come on, no questions from the students. You've mesmerized them. Any questions from? Oh, wait.
Yeah.
There's a question.
Yeah. Yes. Yeah.
So hold it. So with the temperature, does it affect gibberellic content or not? That was the question.
Yeah, actually, from what we saw, it didn't actually, fortunately, because if it had, then we had to restart it again, because we didn't. Because if it had, then we have to restart doing our validation methods and everything, and that would be a lot of work. So yes, fortunately, it didn't show any effect. So, and now it's actually directly for the industrial implication. So now we can go and say to our farmers and BRA that, okay, you don't have to actually store your plant samples when you harvest it, because the temperature doesn't actually show any effect. So yes, that's the answer, I think.
Did you take the gibberellic acid before transporting on?
Oh no, actually
wait. Do you check the gibberellic acid before transporting or not?
No.
Well, answer the question.
Yeah, no, actually we just took the samples and then we transported it, and then we saw if there's a variation or not. If there was any variation, then it could have seen in the result, but yes, fortunately it didn't. Yeah.
Excellent. All right. If there are no more questions, thank you. That was really exciting. It was, look, a zero answer is still an answer. You've ticked that box. You can move on. So thank you, Nathan. Thank you as well. That was
Thank you, everyone.
and everyone who helped you. I'm sure Caroline and others were very helpful. So thank you so much.
Yes, thank you.
All right. Thank you. All right, everyone. Well, thank you. Thank you all, and those that persevered with us online, thank you. I hope to see more of you in person. We need you in person to support our students. This has been really exciting. Thank you so much, everyone. All right, I will sign off and I will say thank you all and goodbye. Thank you.